Alright, our next section we have here is D22, gene expression. This is another HL only section. It's a little longer than the viruses one. Alright, so what is gene expression? And so gene expression is turning genes on and off to affect phenotype. So remember, phenotype is what we see. Genotypes are the genes that you have, the copies of the alleles. But what you can do is you can turn genes on and off, which will then affect your phenotype. And so this can happen in a variety of different ways, which we're going to learn about. The three main ways are transcription, translation, or modifying the protein itself. So when we say regulation of transcription, what do we mean? So we have our DNA, and we have these regions. You can have enhancer regions, you can have the promoter, you have to have the promoter region, and you can also have silencer regions. All three of these can bind transcription factors. So this is DNA. They can all bind transcription factors, which are typically proteins. So these are proteins. And so then they can bind there. So for example, if you bind one here, that'd be like a repressor protein. That would turn it off. If you bind one here or here, if you bind TFs here, these transcription factors are often called activators or activator proteins. And so what they will do is these two will upregulate, whereas this one here will downregulate. You can also have repressors block to the promoter region too, where they would block it. So you can also have a repressor bind to the promoter too and block it. And if they do that, that would also lower transcription. So that's pre-making it, right? So you can't make a protein if you can't do transcription, or you can make more of a protein if you really want to do transcription. 3. Control. So this one is all about, okay, you made your mRNA, now what? Well, you can degrade it at any point. You can break it down. And so some mRNA will last for days, and some last for minutes. And so the ones that last for days will make a lot of protein, and the one that lasts for minutes will make small proteins, a small amount of proteins. And so these are broken down by nucleases. And so depending on how active your nuclease is, nuclease being ace meaning like enzyme cut, nuclease referring to nucleic acid, so you can have it chop it apart. Okay? And so that is another method of regulating how much protein you make. So this one you're going to make a little bit, but you could make a little or a lot depending on how long you wait before you break down the mRNA. The next standard 4 is basically a definition. It's the definition of epigenesis. And this is the development of patterns of differentiation in cells of a multicellular organism. So this is just saying that we can study the different expression patterns in different cell types to figure out how it is. And so the key here to remember is epigenetic changes are not changes to the DNA, they're changes to like tags on the DNA. Okay? So you're not changing the DNA itself, you're just activating or suppressing certain regions. Standard 5. Another kind of vocab, we have 3 words. We have the genome, the transcriptome, and the proteome. And we can see already how these words would be different. The genome is all the genes, and so this one is the DNA. Every cell has all. Every cell has all. But the transcriptome is which mRNA you actually make. So the mRNA is actually made in a cell. And so if you turn some genes off, you won't make them. And then if you degrade some of the mRNAs, you won't actually make all the proteins, which is the proteome is the proteins actually made. And so you can see the only one that is the same in all cells is the genome. These other two are going to be different, which is what leads to epigenetics and cell differentiation. How do we do that? Well, there's a couple of ways to do that. And one of the ways we can get these changes is with methylation. And so methylation is when you add a methyl group to something and it's inactivating it. It's going to inactivate it. And so what you can do is you have cytosine, which is a DNA base, and if you methylate this, you turn it off. And so typically you're doing this in the promoter region. And so you take a C in the promoter region and you methylate it and then you turn it off so it stops transcription. That's one of the ways you can change it. Another one is if you methylate histones, those are the things that make up nucleosomes. If you methylate this, an amino acid of a histone, it causes it to bind more tightly and prevent transcription. The inverse is true. If you acetylate it, acetylation will increase transcription because it loosens binding. And so you have your nucleosomes, right? There's 8 of them. Or sorry, your histones, there's 8 of them in a nucleosome. And then how tightly the DNA is bound is whether it's methylated or acetylated. So in methylation, it'd be very tight, and acetylated DNA would be much more loosely bound. And so you can see how you'd be able to do transcription there. Okay. That was standard 6. Standard 7. Epigenetic inheritance through heritable changes to gene expression. Okay, so now almost all tags are wiped out. So most epigenetic tags are wiped out. Epigenetic tags are gone in sperm and egg. But not all. Most, but not all. Okay? So what this means is that for the most part, the changes can't be passed down. But there are some that can. And so these tags that are from DNA methylation or histone modification, the one we just learned about, some of these can be passed down. And this will affect the offspring. These epigenetic tags are one of the things that make cloning more challenging, right? And so I'm actually going to skip to standard 9. I'll come back to 8. And so since most are removed but not all, you can get some weird effects. And the example is our liger versus our tigon. And so if you have a male lion and a female tiger, you get a huge liger because they have both of their size genes are on. Check, check. in a tigon, you have a female lion and a male tiger, and this one is off and this one is on. And so what you have is it's much smaller because the female lion has an epigenetic tag that blocks the growth hormone that then causes it to be smaller. And it's because lions normally have more than one cub at a time, whereas tigers typically only have one or two. And so it's like a behavioral difference that has led to this epigenetic tag that is like found in them, but when they interbreed like this, it doesn't pass down correctly. All right, so other things. How do we affect methyl tags? So one of the things that can affect your methyl tags on DNA is air pollution. So air pollution can have an effect on your methyl tags and cause them to be upregulated, which what happens is you're actually causing issues. So what kind of issues are you causing? It causes an overreaction of the immune system. And so this can lead to damage to lungs, lung cancer, cardiac disease, and so your immune system basically is like overworking, and it causes it to damage it. And this is because what you've done is you have activated genes that were not supposed to be activated, and so you have these methyl tags that are not working like they should because of the air pollution. How do we know about methyl tags? And how do we know about the environment effect? Well, we know about environment effects. We know about this because of what are called monozygotic twin studies. And so these are when you study twins to look and see how the environment affects them. And so it's the environmental effect because these two are they should be identical, but they're not identical. And so what you do is you study their DNA and you specifically look for the methyl tags, for the methylation, and you look for differences between them. Because it's identical DNA, therefore the methylation is one of the only differences between them. Alright, so the last standard is all about these two different pathways, and we actually learned about this again later on, but one of them is insulin. So insulin affects gene expression. And so, spoiler alert, if you have not learned this yet, but it is, this one here, right, is a peptide hormone, and it's a transmembrane receptor. And so what it does is it causes a cascade pathway to upregulate creation of the GLUT4 transporter, which is a glucose transporter. And so then you take in more glucose. So because insulin attaches to this receptor, it causes a cascade pathway, which creates more glucose channels by triggering transcription factors to create more GLUT4, which is a glucose transporter. The other one is lactose. And so lactose will enter the cell in bacteria, and it activates what's called the lac operon. And so it binds to another molecule to form a transcription factor. So the transcription factor is lactose plus a molecule, and a transcription factor, right, is going to act as an activator in this case, and it's going to activate the lactase gene, which makes sense. You only want to make lactase when you have lactose, and so that's what this gene does, is it causes it to make lactose when you have, it causes you to make lactase when you have lactose present. And that is gene expression.